What is that?
It's a firefly.
No, it's obviously a fairy.
I think it might be a firefly, but why is it glowing?
Well, guys, it is glowing because of a molecule called luciferin.
But what is luciferin?
At the molecular level, firefly light comes from a small molecule called luciferin. But luciferin is not just one molecule. Different organisms use different luciferins to produce light. This is luciferin, and they're all luciferin. They look very different, but they actually share some important structural ideas. Even though these luciferins have different shapes, they share several key structural features that they make light emission possible. First, all luciferins contain extended conjugated system, which allow electrons to move across the molecule. Second, they include heterocyclic structures with atoms like nitrogen or sulfur, which help regulate electron distribution. And finally, luciferins tend to be relatively rigid and partially planar, which reduces energy loss as heat and favors light emission. When energy comes to these two structures, they behave very differently. The flexible one wiggles and releases heat. The rigid one glows. Now that we've seen the shared structural features of luciferins, let's bring our focus back to firefly luciferin. Firefly luciferin is especially well studied, not just because it produces light, but because it does so in a highly controlled and consistent way. The structural features we highlighted earlier, conjugation, heterocycles, and rigidity, define how this molecule handles energy at the molecular level. Together, they make firefly luciferin a reliable system for studying bioluminescence. In the next part, we'll take a closer look at what happens when firefly luciferin is placed into a biological system.
Alright, so why do fireflies glow? To understand this, we must first take a look at firefly anatomy. Within the abdomen of a firefly sits a light-producing organ called a lantern, which contains specialized cells known as photocytes. It is within these cells that bioluminescence, the chemical production of light, takes place. And actually, fireflies are perhaps one of the most well-known examples of this phenomenon in nature. Within the photocytes, the luciferase enzyme is localized into compartments called peroxisomes, where the reaction actually takes place. A molecule called D-luciferin, or also referred to as beta-luciferin, is a stable, non-toxic compound that is acted upon by luciferase. And when luciferin combines with oxygen, ATP, calcium, and magnesium, luciferase catalyzes a reaction that converts beta-luciferin into a whole new molecule called oxyluciferin. Subsequently, AMP, CO2, and pyrophosphate are released as byproducts. The result is the production of visible light in the yellow, orange, and red hues, depending on the species. And unlike a typical light bulb, very little energy is lost as heat, making it an exceptionally efficient reaction. The firefly controls its flash by regulating the oxygen supplied to its lantern. Because fireflies do not have lungs, oxygen travels through tiny tubes called tracheoles. When no light is needed, oxygen entering the lantern is absorbed by the mitochondria and kept away from the luciferin reaction. When the firefly releases nitric oxide, it binds to the mitochondria instead, which frees up oxygen to flow into the lantern and trigger the bioluminescent reaction. Since nitric oxide breaks down rapidly, the flash is short and precisely controlled. Notably, the luciferase enzyme plays a dual role in the system. In addition to catalyzing light production, it also functions as an acyl-CoA ligase, participating in the recycling and biosynthesis of luciferin itself. After the bioluminescent reaction, the spent oxyluciferin is converted back into a usable luciferin through a dedicated recycling enzyme, ensuring the system can continue to produce light.
In order to explore how exactly luciferin's bioluminescence reaction is so efficient, we must look deep inside a firefly's lantern, where its bioluminescence reaction takes place. This phenomenon is known as cold light efficiency, which minimizes heat production while maximizing energy conversion into visible light. Firefly bioluminescence processes were originally thought to have a quantum yield of nearly 100% of energy converted to light, but recent studies have found that the quantum yield is actually around 41%. This number is much lower, but still quite impressive compared to the 10% yield of typical incandescent light bulbs. Inside of the active site of the luciferase enzyme, 1,2-dioxydenone is produced as a key intermediate responsible for the visible light released. 1,2-dioxydenone is produced from the initial luciferin substrate with the help of magnesium, ATP, and oxygen. It is a very high energy intermediate. This four-membered ring is so unstable that an electron from a more electron-rich part of the same molecule is shared to it, leading to two bonds breaking. Once these bonds are broken, the electron returns to its owner in a higher energy orbital this time. This leads to the release of an excited state oxyluciferin and a carbon dioxide molecule. This additional energy must be released, but since the luciferase enzyme is holding oxyluciferase tightly, this energy cannot be released in the form of movement or heat. Instead, it is released in the form of a photon or visible light.
One of the most fascinating aspects of bioluminescence is that it did not evolve just once. Scientists estimate that bioluminescence has evolved independently more than 40 times across different evolutionary lineages. This is the example of convergence evolution, which occurs when unrelated organisms independently develop similar traits because they face similar environmental pressures. A strong example of this is seen in jellyfish. The bioluminescence in species such as the crystal jellyfish evolved separately from the light-producing systems found in fireflies or in dinoflagellates. In jellyfish, light production involves a calcium-activated photoprotein called ecorin, which works together with green fluorescent protein, also known as GFP. This system is structurally and chemically different from the luciferin and luciferase systems used by fireflies and dinoflagellates. Marine environments create strong selective pressures such as predation and communication in darkness, and survival in deep or low light waters. Because light travels efficiently through water, producing light becomes an effective survival strategy. As a result, different organisms independently evolved glowing abilities even though the molecular mechanics behind the light are different.